Hello everyone, this is the first lecture of veterinary epidemiology. So in this lecture we will be discussing the basics of veterinary epidemiology, what is veterinary epidemiology, its definition, its objectives and a few more points regarding epidemiology, very basic points. Okay. So epidemiology, before moving on to a definition of epidemiology, we should know that epidemiology derives from three Greek words, epi, demo and logo. Epi means upon, logo means demo means people or population, logo means discoursing or study. So epidemiology is the study of disease in a population and of factor that determine its occurrence. So epidemiology is the study of disease in a population. We will study a disease in a population, and we will also study the factors which affects the in which determine the occurrence of disease in the population. We will study those factors also. That is what is called epidemiology. Okay. Now, basis of any epidemiological study is a population. So, any epidemiological study is dependent on population. Okay. Okay. Now, there were various scientists who gave different definitions. So, one of the definitions is this. So, this is the standard definition. This is the standard definition of epidemiology. So, the standard various scientists were, one of them were Schwab. So, according to him, the research discipline concerned with the distribution and determinants of disease in a population. We cannot do anything. We just have to mug up these definitions. There were three or three more scientists. For example, Helfin. So, she gave the definition of epidemiology as the study of pattern of disease. The other scientists were McMahon and Pug. So McMahon and Pug, they gave the definition of epidemiology to humans, and there was one more scientist, Norman D. Levine. So according to him, epidemiology is ecology. Epidemiology is the ecology with its mathematical and medical flavor. So these are the basic definitions. We have to just mug up these definitions given by all these four different people. Okay. Let me keep these on mind. We will be focusing on this page. Now there are few terms. So here I have written three terms, but we will be discussing four terms. So first term is epidemic, then epizootic, then epioornithics. So epidemic, outbreaks of disease in human population. Epizootic, outbreak of disease in animal population. And epioornithic, outbreak of disease in avian population. Because ornitho means bird, zoo means animals. Okay. Okay. So if we try to understand, there is one more term that is called endemic. So endemic is any disease. Endemic is any disease that which is present in a particular area at a constant rate, which is always prevalent in an area at a constant rate in spite of the strict preventive measures which have been taken by the authority of that place. Okay. Okay. So now we'll move on to the objectives of epidemiology. There are five objectives of epidemiology. First is determination of the origin of a disease whose cause is known. So this point basically means that if there is any disease whose etiology is known. Okay, so this is the second lecture, detailed lecture on epidemiology. So here we will be discussing the five objectives of epidemiology. There are five objectives of epidemiology. So first is determination of origin of a disease whose cause is known. So here what we are trying to do is we have a disease whose etiology we know, etiology as cause we know, but we do not know how the disease originated in a particular population. So that is what we will be able to trying to find out with respect to the first objective of epidemiology. Okay. Okay. So we can understand this with the help of examples which was given in the book of our college. So teacher's notes, that is. So that was suppose that there was a postman. There was a postman who was showing the lesions of cutaneous anthrax on his neck region. So what happened? What was done, that it was traced back. His actions was traced back, and it was found out that that postman used a scarf which belonged to his son, and his son worked in a tannery. And if you don't know, tannery is a place where the skin and hides of the animals are handled. So that from there he got the lesions of cutaneous anthrax. Okay. Now second objective epidemiology is investigation of a disease, or investigation of and control of a disease whose cause is either unknown or poorly understood. Alright, so what does this mean? It basically explains us that if there is any disease whose etiological factor we don't know, or we do not know the etiological factor of the disease, so what we can do is we can basically try to investigate about that disease and try to control the disease. So example with respect to this can be, I haven't written example here, but it is written in a college book. Let me show you. So this is a book of a college. So here they have given the example. See, this is the second objective and these are the examples. So Examples can be Edward Jenner. He eradicated smallpox using the using the virus of cowpox, or using the disease cowpox. Okay, because back then the causative agent, that is etiology of smallpox, that is pox virus, was not known. Okay, so that is the thing. And another example is that this what do we say? Contagious bovine pneumopneumonia, CBPP. It was eradicated from US in 1872, even before Mycoplasma mycoides. Its causative agent etiology was discovered or identified. Okay. All right. Next, we have the third objective, that is acquisition of the information of ecology and natural history of a disease. So there are various terms. First, we should understand these terms. There is natural history. So there are two terms: natural history and natural history of a disease. There are two terms. Okay. So we try to describe these two terms. Natural history basically means aggregate of all facts regarding plants and animals. But if we talk about natural history of a disease, so natural history of a disease means that how a disease starts, initiates, and how it progresses without any human intervention. For example, a disease, a population or a herd acquired a disease. So if human will not intervene in terms of its prevention, control, or treatment, then what will be the end result of the disease? That can be in three form: recovery, death, or invalidism. So that is the meaning that is explained by, that is the meaning of a natural history of a disease. Now next we have other terms. Ecology will discuss just right now. So agent, host, and environment. So these three are the factors which are actually involved in the occurrence of a disease. Okay. So we'll discuss later that this was one of the shortcomings of Koch postulate. In another lecture we'll discuss that this was one of the shortcomings of Koch postulate, because he only focused on agent as the causative agent of a disease. Okay. But we need agent, host, and environment, all these three factors, if we want any disease to take place. Otherwise that disease will not take place. Okay. So what is a host? First we'll discuss host. So host is an animal or organism. Host is an animal that is susceptible to and can be infected by an agent. Easy. Agent, any factor whose either presence, excess presence, or absence leads to a disease. And agents are of three types: physical, biological, and chemical. Okay. Physical, biological, and chemical. So we talk about biological agent and pathogen, parasite, chemical, toxin, acid, bases, physical, radiation, heat, cold, all of this. Okay. Host we will discuss now. Environment. Environment, that is aggregate of all the conditions, aggregate of all external conditions that affect the life, development, and behavior of the organism in the society. Very easy. Okay. So aggregate, that is collection of all those external conditions, because environment is actually talking about external environment. Okay. So aggregate of all external conditions, and those conditions they are actually that important that they regulate the life cycle, they regulate the life cycle, they regulate the behavior, they regulate the development of that particular organism in the society. Okay, life cycle is not a correct word because we are not studying parasites here. We are not talking about parasites here. Okay, obviously. Now we'll discuss ecosystem and ecology. So ecosystem is actually, what is an ecosystem? Ecosystem is a community of organisms which is formed by the interaction between ecosystem is a community which is formed by the interaction between biotic and abiotic components and the flow of energy and nutrients between them. Okay, that is what ecosystem. Then next is ecology. Ecology is study of ecosystem. Very easy. Okay, and fourth objective is planning, monitoring, and assessment of a disease control program. So we talk about planning, monitoring, and assessment of a disease control program. Obviously, when we have understood everything regarding a disease, we have collected all the data which is required regarding a disease in the first three objectives. Now we need to plan a disease control program. Okay, so there are two important terms here which you need to study. First is monitoring and second is surveillance. Okay, I hope this is clear. Yeah. First is monitoring, second is surveillance. So if we talk about monitoring and surveillance, and monitoring is the process of making routine observations with respect to health and productivity, and it also includes the recording, transmission, or collection and transmission of these observations and transfer of that recording or data to authorized authorities. Not concerned authorities, concerned parties. Okay. So for example, regular meat inspection in an abattoir. So now I will explain the meaning of monitoring. So monitoring is the process of making routine observations. So we can understand this with the help of this example. Regular meat inspection is an abattoir. Abattoir, that is a slaughterhouse. Abattoir, that is a slaughterhouse. So we are regularly inspecting the meat quality in an abattoir or slaughterhouse. So that is what is called monitoring. So what do we do in monitoring? In monitoring, we make routine observation with respect to the health and productivity of a particular population or a particular food item the quality of the produced food item, but here we are talking about population epidemiology, that is why we are talking about health and productivity. So the process of making routine observation with respect to health and productivity, and then also the recording of that observation, and then transfer of that observation to the concerned parties or concerned authorities, that is called monitoring. Okay. Now if we talk about surveillance, then surveillance is the most intensive form of data collection, as it involves data recording, data transfer to the concerned authorities, as well as the analysis of the data. Okay, so that is the meaning of surveillance. All right, so we'll take the example to understand surveillance with the help of this example. This is the example. It is again given in our college book. So first is, suppose that we identified an animal, say a cattle, which was roaming the streets, and we identified that that cattle was suffering from the lesions of TB. That was showing the signs of TB, the symptoms of TB. Now what we did was we traced back the origin of that cattle, that farm of origin of the farm from which the cattle has been left out or released. And then what we did, we reached that farm, and we then used tuberculin test, because this is an example of TB. We used tuberculin test on all the other animals of that herd or farm, and then on the basis of the result of the infection, we actually planned a control program, if we need a suitable control program or not. All right, let's move ahead. So this is the last objective, that is assessment of economic effects of a disease. Obviously this is the last objective of epidemiology. It is the fifth objective. If we have made a disease control program, we need to be aware and we need to keep in check how much is the cost involved, how much is the benefit, how much are the losses. Okay, thank you. Hello everyone, so this is the third detailed lecture on veterinary epidemiology, and the topic is factors influencing the occurrence of livestock diseases, part 1. Alright, so there are two terms which you already know, health and disease. So these two definitions were given by WHO. So you need to learn these two definitions, exact definitions. Okay, so what is health? Health simply can be defined as the absence of disease, but it is more than absence of disease. Health is a state of complete physical, mental and social well-being. Okay, and not merely an absence of disease according to WHO. Perfect. Now, disease condition of body or organ in which its function are disrupted or deranged. Very easy. Now, in the previous lecture, we discussed about post postulate. I told that in the next lecture or later lecture, we will be discussing about post postulate. So, they were given by Robert Koch, and there are 4 in number. Total 4 postulates are given by Robert Koch. Okay. So, first postulate says that the microbe must be found in every case of the disease. Second, the microbe must be isolated from the diseased individual and grown on a pure culture. Third, the cultured microbe should cause the same disease when it is introduced into a susceptible, healthy susceptible individual. Okay, and fourth postulate is that the microbe must be re-isolated from the experimentally infected animal host and found to be identical to the original microbe. Can't do anything. These are just the 4 postulates. Now there are 2 most important shortcomings of those past postulates. Koch postulate, two important drawbacks. First, they overlook the effect of Hello everyone. So we are discussing the Koch postulate, shortcoming of Koch postulate. So there are 2 most important shortcomings of Koch postulate. That is, they overlook the effect of environmental factors in the causation of a disease. What this shortcoming is basically trying to say is that according to Robert Koch, the most important factor with respect to a disease was only and only the presence of agent. But this is not true. And why is this not true? Because now we know, according to epidemiological principles, there is a triad that is called epidemiological triad, and all the 3 factors, that is host, agent, environment, should be present in order to cause a disease. Okay. A second postulate was these postulates not applicable to non-infectious disease. He was always talking, those postulates are always talking about microbe. So these were not applicable to non-infectious diseases. All right. Now we move on to a very important topic, that is called determinants of disease. Okay. So what is a determinant? First, we need to understand this. These are the causes or risk factor associated with the increasing the probability of a disease. Alright, let's now simply understand this definition. So determinant means any factor which determine the presence or absence or occurrence of a disease. Okay. So in determinants, what happens? These are the causes or risk factor associated with the increasing the probability of a disease. So if these determinants will be present or absent also some cases, then there will be higher probability or there will be higher risk factor, there will be higher risk that there will be higher chances of probability that this disease will take place. Okay, Okay. Now we'll discuss the classification of determinants. What are determinants? These are the causes or risk factors associated with the increasing in the probability of a disease. Alright. Next we have classification of determinants. So first we have intrinsic and extrinsic determinants. So intrinsic determinant, factors present in the individual and are any characteristic of that individual. Obviously intrinsic present within. So any characteristic which are found within the individual and that characteristic is acting as a determinant. That is called intrinsic determinant. We understand this with the help of an example. So examples are these: genetic constitution, genotype, age, breed, sex. All of these are the different genotypic, also intrinsic determinant. Obviously, it's present inside within the individual. All right. Now next we have extrinsic determinants. So these are the determinants or factors which are present outside the host. So for example, climate, food, transport, microbes, all of this. All right. Now we will understand intrinsic and extrinsic determinants with the help of example. So an example, that is bovine rotaviral diarrhea. So there are two intrinsic determinant and two extrinsic determinant or factor for this disease. Okay. So the intrinsic determinant are age and immune status. So what does this mean? This means that as we know, rotaviral diarrhea or rotavirus affects young calves. Okay. Now immune status, obviously if the host will be immunocompromised, calf will be immunocompromised, then that calf will easily catch the infection. Now what are the extrinsic factors? So why these were the intrinsic factor? Because age is an intrinsic factor, as we already know. Immune system status is also an intrinsic factor, it is present within the host. Okay. Now extrinsic factors, so first is winter season and second is the presence of virus itself. So as you already know, if the virus will be not present, if virus will not be present, then the infection will not take place. But winter season, rotavirus more commonly causes disease in the winter season. Okay. Alright. Next we move ahead. These points. Next we have primary and the secondary determinant. So we discuss intrinsic and extrinsic determinant. Next is primary and secondary determinant. Okay. So primary determine the factors whose variation exert a major effect in inducing diseases. Okay. So primary and secondary diseases factors. So primary determinants are the factor that is the main causes of a disease. As we have written here, these are the main causes of a disease. Okay. The factor whose variation exert a major effect in inducing disease, and these are the main causes of a disease, that is called primary determinant. Okay. So primary determinant, let us understand this. Primary determinant are the determinants or factors in which if variation took place, then they will exert a major effect in inducing the disease. Okay. That is what is called primary determinant. So example can be exposure of a dog to canine distemper virus. Obviously if a dog is exposed or canine population exposed to canine distemper virus, obviously that is the primary determinant that population of the dog will or may develop CD, canine distemper. Okay. Alright. Now next we have secondary determinant. So these are the predisposing or reinforcing factors. Okay. So these are the predisposing or reinforcing factors. Example can be sex is a secondary determinant of CHVI. Okay. Let us try to understand this. So secondary determinants are the factors which are the predisposing or reinforcing factor. That is, these help these reinforce or these what do we say? What term do we use? These reinforce or predispose, or these make that particular condition worse for that particular animal or host, so that that animal or host can catch the disease easily. Alright. Now the example is sex is a secondary determinant in CHVI. CHVI is a condition that is canine heart valve incompetence. This is a abbreviation Shai made. That is, I don't think so that is a standard abbreviation. So canine heart valve incompetence, so obviously as the name suggests, that is something related to valves of the heart. So male dogs are more likely to develop this disease than female dogs. That is a secondary determinant. Okay. Third determinant we have the epidemiological triad. So it includes agent, host and environment. Okay. So this is what the epidemiological triad looks like: host, agent, environment, HE. Okay. Okay. Or AHE extension. Okay. Now we will discuss host determinants. So host determinants, first is genotype. Okay. So we talk about genotype, then it is the genetic constitution of an animal, that is called genotype. Okay. And the disease in which the genes are the aberration in the genes are the primary determinants that are called genetic diseases. So what are genetic diseases? Genetic diseases are the diseases in which the aberrant or alteration in the normal gene is the primary cause of the disease. For example, hemophilia A and B in dogs. Alright. So next we have age. First was genotype, second we have age. So age means some diseases are more commonly found in young population, some are found in older population. Alright. So the diseases which are found in younger population, the example are rotaviral diarrhea, Japanese encephalitis. Older population, protozoan rickettsial, that is parasitic diseases or cancer, and neoplasia. Now next is sex. So that it has various two types of subdivisions, that is hormonal determinant and occupational determinant. So hormonal determinant, there are two examples. So we took two female dogs, adult female dog. Okay, what we did, we spayed one of them. That is, we performed oviriohysterectomy on that particular dog. We removed her reproductive organ. So now what will happen? Due to hormonal imbalance, some hormonal imbalance that we do not need to discuss here, and I also don't know obviously. So I'm a third year itself. So memory carcinoma is more common and seen in neutered female dog. Okay. So neutered bitches or female dog more commonly seen is memory carcinoma, that cause malignant cancer of mammary gland. And diabetes mellitus is more commonly seen in normal female dog. Okay. Okay, next we have occupational determinants. Occupational determinants, so this is also included within the gender or sex. Okay, so occupational determinant, it basically means that due to a certain gender of a particular animal, that animal is used for a certain particular kind of work. Okay, so example can be male dogs. So in the hilly areas of snowy areas like Arctic and Antarctic region, husky dogs, male husky dogs are used for when they are used for hunting. So they directly come in contact with the mosquito in the forest. So they develop the infection heartworm more common than female dogs, female husky. Okay. Now other host determinants are size and conformation and coat color. So we talk about size and conformation. So we will find out that hip dysplasia is more commonly seen in larger breeds of dog. Okay. Cows with small pelvic outlet in comparison to their body size suffer from dystocia, that is difficult birth. Alright, coat color. So this can be understood by a simple example. If any animal is, coat color of any animal is white or grey, then that animal will suffer more commonly from skin cancer or squamous cell carcinoma. Malignant tumor of squamous cells. Okay. And second we have, if an animal is of brown or black or any dark color, then that animal will have very less chances of suffering from squamous cell carcinoma because melanin prevents or protects the skin from UV rays. Okay. Okay. Now, next we have another chapter, that is factor influencing occurrence of livestock diseases, part 2. Okay. So we have discussed host determinants, now we will discuss environmental and agent determinants. Okay. So environmental determinants include location, climate, husbandry practice and stress. 4. Okay. First we have location. So location, obviously, location basically depends, we are talking about location with respect to the climate, not climate, with respect to their, what do we say, vegetation. So the large amount of vegetation, large and better vegetation, larger will be the host and larger will be the infections. Okay. For example, CCPP and CPP are found in the more commonly in the hilly area of Assam. Easy. Okay. Now climate. So there are two types of climate, macro and microclimate. Macroclimate, these are the normal components of the weather to which the animals are exposed. Okay. For example, rainfall, temperature, humidity, radiation, wind. Alright. Now microclimate, these are the climate that occurs in a small defined space. Okay. The climate that occurs in a small defined space, that is called microclimate, and the example can be within a few millimeters of plants and animals or in an animal house. So let's discuss this in detail, little bit detail. Okay. So we have, with respect to microclimate, we will take the example of animal house. So suppose that we will take two species in example to understand. So those two species, one will be poultry and second will be pigs or swine. So what we did was we visited an animal house, animal house, which was poorly ventilated, and there was high concentration of ammonia gas. So in the case of poultry, what will happen? That will lead to keratinoconjunctivitis, simple blindness. Okay, in poultry. But what will this lead to in pigs? So this will lead to turbinate atrophy. What is turbinate atrophy? Turbinate atrophy is the turbinate bones are present inside the nasal cavity. So those bones will shrink in size. Okay, so that is the thing. This is not important. You know this. Alright. Now third environment determinant is husbandry practices. So husbandry practices, as we already know, there are three most important husbandry practices: housing, diet, and management. So management, we will talk about management first. So management, obviously, there should be regular deworming, colostrum feeding, regular vaccination, all of this. Diet, there should be balanced diet so that animals remain or live free from diseases. And housing, floor should not be slippery, floor should not be too slopey, all of this. There should be ventilation in the houses. Alright. Now we will discuss stress, the fourth environmental determinant. Okay. So if you talk about the fourth environmental determinant, then the stress, that is a stress, so it is a sum of biological stimulus that leads to a disturbance of homeostasis. Okay. So any biological stimulus that is disturbing the homeostasis, that is constant internal environment of the body, that is included in the stress. Alright. Now all the factors which produce stress are called stressors. Very easy. Now the important topic is this also. With respect to stress, in lab animals, to study the effect of stress on lab animals, there was a hypothesis given, that was Seeley's hypothesis or general adaptation syndrome. Okay. So it describes the effect of stress on lab animal. Okay. Now it is divided into three phases. Listen to me very carefully. Seeley's hypothesis, I don't know whether this is important or not. I just read it because that is given in the book, a college book. Okay. So there are three parts or phases of Seeley's hypothesis of general adaptation syndrome. So those are GAR, that is general alarm reaction, phase of adaptation or phase of resistance as it is also called, and phase of reaction. Phase of reaction leads to that. Okay. Now general alarm reaction, it occurs 6 to 48 hours after exposure to stress. Phase of adaptation or resistance occurs 40 hours after exposure to stress. And what happens? In general alarm reaction, there is atrophy of liver, spleen, and lymph node. In phase of adaptation resistance, there is both hypertrophy and hyperplasia of adrenal gland. Only adrenal gland, or we can say adrenal medulla also. Okay. Alright. Next we have agent determinants. Agent determinants, so we will discuss various important agent determinants. The first is virulence and pathogenicity. These two terms are different. So pathogenicity is the ability of an organism to cause disease, and virulence is the ability of an organism to cause disease in terms of its severity. Okay, how severe is the disease, that is told by virulence. So virulence, the formula is number of clinical cases upon number of the population affected. Okay, infected, not infected, affected. Okay, and pathogenicity, number of clinical cases on population exposed to infection. Okay, we cannot do anything, we just have to mug up these information or these formula. Okay. Alright, next agent characteristic, agent determinant is gradient of infection. So what is the meaning of gradient of infection? So it is the range of responses produced by an organism when it is exposed to a particular infectious agent. Okay, so gradient of infection is the range of responses which is produced by an organism when it is exposed to an infectious agent. Okay, what are different types of responses a particular individual or organism or population will produce when they are exposed, or when that individual is exposed to a particular infectious agent or pathogen. So there are three gradients of infection. First is inapparent or silent. Second is subclinical, and third is clinical. We will discuss clinical first. Clinical, obviously there will be easily we can diagnose with the help of clinical signs. Now we have two types in clinical infection, mild form or abortive reaction and frank clinical form. So mild form or abortive reaction will not be able to easily diagnose the case because there will be less infection, less clinical diagnostic signs or clinical signs. In frank clinical form, you will be able to identify the disease very easily because of the clearly visible clinical signs. Okay. Okay. Now next we have inapparent or subclinical. So listen to me very carefully. So inapparent or subclinical, both of these, the common characteristic are that there will be no visible clinical sign. Both type of affected animals, there will be presence of antigen antibody in the serum, and the difference is in inapparent infection there will be no loss of productivity, and in subclinical infection there will be loss of productivity. Okay. Alright. So next agent determinant, we have outcome of a disease. Okay. So outcome of a disease, there can be three outcome of a disease. First is recovery, death, or invalidism. So recovery, we know the animal has developed an immune response. Okay. Death, animal was not able to survive. And invalidism, third is invalidism. Invalidism, that is development of chronic infection. Okay. And this leads to the formation of a carrier. Okay. So what is a carrier? A carrier is an individual organism or organism who sheds an infectious agent without showing any clinical sign itself. Okay. So any organism which is actually shedding an organism, that is releasing or discharging organism in its body, discharges or excretion without showing any clinical sign itself, that is called a carrier. Okay. There are two types of carrier, incubatory and convalescent carrier. Okay, incubatory carrier and convalescent carrier. So incubatory carrier, as the name says, incubatory. There is something to do with incubation period. Okay, so incubatory carrier is the animal or carrier that excretes the agent during the incubatory period of the disease. Okay, during incubation period, that is, what is incubation period now? The period from the infection, that is entry of microbe into the body, to the development of clinical signs. Okay, so incubatory carrier. So example can be dogs. They shed rabies virus in their saliva 5 to 15 days before the development of clinical signs. Okay. Next we have convalescent carrier. So it carrier animal that sheds an agent when it is recovering from that particular disease. Okay. For example, pigs shed leptospira in their urine for 6 to 12 months. Okay. Okay. So next we have latent infection. Obviously, latent infection means there will be no clinical sign, but an infection will persist. Okay. And last is microbial colonization of host, that is the fourth agent determinant. Okay, so there are two types of microbial infection, vertical and horizontal. So vertical from mother to fetus, horizontal between the animal of the same generation, either through direct or indirect contact. Okay, so what is the meaning of vertical transmission? Vertical transmission means that is the microbe is being transferred from one generation to another with the help of placenta. Okay, that is through birth. So vertical, from mother to fetus. And horizontal, that is from within the same generation. That can be direct, indirect, opportunistic, endogenous or exogenous. Alright.